applied to many aspects of our body. But everything involving the brain and spinal cord is a far greater challenge for those of us who are trying to apply this to precision medicine, in part because the brain and spinal cord are hard to get to. So we began a program, a spin-out of the Packer Center, now about 2.5, 3 years ago, called Answer ALS. It's a large collaborative team. And it really began, if we're honest, with an initial endeavor from the NIH, where they were trying to help us work as basic and clinical scientists to rigorously obtain data, mostly from cancer, but a little bit from neural cells as well. And the approach that Anthony mentioned about how do we uncouple all the patients that look alike to us to individualized patients, this was particularly important for neurologic disease because, and you saw sort of a version of this before, when we see patients with ALS, by the way, this could easily be Alzheimer's or Parkinson's, basically sporadic diseases with not a single genetic cause. Most of these patients are variable, and quite honestly, if I have a patient with one mutation that I later learn has a mutation, that patient with a known genetic mutation looks no different from a patient who has no known genetic cause. We can't separate the patients very readily by our clinical exam, at least with the tools that we've been doing for almost 150 years. No one's come up with a way to discriminate patients based on their clinical phenotype for this kind of disease. And the reason that's important is because knowing what starts the disease is in some hands the best way of developing a therapy. And the lessons learned that we want to apply to a disease like ALS come very well from non-neurologic diseases. So when I trained in medicine, when I began many years ago, if someone had a breast cancer or prostate, we'd look under the microscope and we'd say, oh yeah, that's cancer. Patients were treated, and we routinely failed, quite frankly, with many drugs because they wouldn't treat all patients equally well. But of course, that's the tissue from the patient. And if we can analyze that tissue and do the molecular approaches that you're hearing about from Dr. Rosen, we can begin to say, oh, wait a minute, there's a different kind of breast cancer or prostate cancer. There's an estrogen responsive type cancer. I'm not going to go into the particular science, but the point is that by having that tissue, we can begin to ascertain and discriminate different pathways. And by doing that, and then the next patient comes in, we can test and say, oh, you have an estrogen, you're one kind of breast cancer or prostate cancer or melanoma, because we can biopsy all of these, you get a certain drug and it works in you, it won't work in the rest of the patients. That's bad for the rest of the patients, but it's very good for the one patient or a subset of patient that it works in. And that doesn't happen in neurologic disease. That approach, which has been quite successful, not completely successful, but very successful in general in medicine, doesn't work in neurologic diseases. And the reason is because when my patient comes to the clinic, I can't ask them, I can't say to my neurosurgeon colleagues, could you come up over here and numb up their skull and take a chunk of their brain out? Because your brain doesn't regenerate. Your spinal cord's the width of my finger, it's about that long. If I take a piece of your spinal cord out, you're permanently paralyzed and you'll have permanent pain because of the circuitry involved. We cannot sample the nervous system the way we can sample other parts of the human body. Therein lies a problem. How do we then ascertain if the clinical exam isn't teaching us much, and we've learned over many years we can't look at blood and other non-CNS tissues, how do we begin to figure out these diseases and sort through them? And that's actually where, so this is a text slide, it's a little texty, but I'm going to explain graphically. In fact, maybe I'll just move to that. I'll stop here for a second. Answer ALS is a program built really around the ability to generate brain cells, actually generate stem cells, and I'll tell you more about that in a second, from patients' blood. The technology of stem cells, we've all heard about stem cells. On one hand, people think it's a great therapy, but actually many of us that try to understand disease think it's a great platform to understand human disease. Because I can take a patient's blood sample, a simple sample of their blood, with today's technology, and with the right colleagues and collaborators, I can convert that blood, I, by the way, is a royal we, it's not really me, can convert that blood into stem cells, which are then converted into brain cells. And in so doing, we essentially generate the biopsy of the brain. Now, having that technology means we can do what's been done in other diseases. And so Answer ALS is a program to collect 1,000 patients nationwide, not just at Johns Hopkins, the leading centers that have high density of ALS patients. And we're going to follow them, just like Dr. Rosen mentioned. We're going to get a lot of clinical data, because we're going to use that data in the end. We're not only going to get the usual clinical data that happens at any typical clinic visit for patients, strength, we'll take some blood, we'll see how well they breathe, things that are often done in this kind of neurologic disease. We've actually even developed a version of what might be called telemedicine. We've developed apps for iOS, Apple devices, as well as Android devices. So when they go home, every day of the week, we can collect more information, as long as they're willing to enter data and do some games, which I'll show you about. We can learn about their motor activity, their breathing, their speech, even their thinking from these apps. So we can get a deeper density of clinical data. Now, we don't know if that's going to answer that personalization. We'll separate. In fact, I doubt it, but it will help us when we do the next step. So the next part of this process is, I'll go to that now, is that so patients are seen in clinics, and the clinics are actually at Johns Hopkins, they're at Emory University, they're at Mass General, they're all distributed around the country. I'll show you that in just a second. Those patients' blood's collected, and then the blood is converted into these cells called iPS cells, induced pluripotent cells. That's a term, the newer way that we make stem cells from patients' tissues. You can actually make it from fat cells, from skin, but it turns out the most reliable way today is to make it from blood. And the most uniform and stable way. So we make it, and our colleague at Cedars-Sinai, this is all done on an industrial scale. There is no endeavor anywhere in any company on this scale. It's all done uniformly with high quality approaches. And then those cells get analyzed in the most complete biological way possible. We do your entire genome, your DNA. Your DNA actually codes for RNA. The entire RNA profile of the brain cells are done. RNA codes for proteins. We measure all of the proteins in those brain cells. Proteins affect biochemistry, the metabolism of cells. We do a complete, what's called metabolomic analysis. In fact, we even go one step further. We grow them. One of our colleagues at Gladstone, UCSF, can grow those cells and actually watch how well they grow their nerve processes, how well they live. So this is an enormous amount of data. And that data, the idea of having all of this clinical and biological data has to be integrated. You're going to hear from Ernest Frankel in a few minutes about how do you integrate this data? Because the idea is to take all of this data, not just their genes, but all of the things downstream of genes, to come up with essentially subgroups of patients that they're defined in this largely sporadic disease, ALS. And this same process actually can be done for any neurologic disease. And then we have to integrate the data, and Ernest will tell you a little bit more. We've actually been doing this. How you integrate, how do you use all of that data together? 6 billion data points approximately per patient will be generated, actually are being generated. How do you integrate that to come up with a signature? The idea is if you come up with a signature, maybe there in fact are 5 different groups of ALS patients. Again, this applies to the diseases. You'll prospectively be able to, in the future, when a patient comes into the clinic, you'll know to measure for these, say, 20 different markers, and we'll say, you're in group A. And if you're in group A, that means from studying those pathways, those cells, you deserve to get drug X, aspirin, a heart drug, whatever the drug might be. And now we personalize the approaches to medicine for ALS. So this project's now been underway for about 1.5 years. There are centers largely distributed around the United States. I told you I grew up in New Jersey. You can see this is a little centric to the East Coast, because we all know the real bulk of important medicine comes from the East Coast. From MIT, from Johns Hopkins, from Harvard, all second built, of course, to Johns Hopkins. But we've included centers really around the country. And we want to distribute them, really want access. Not all patients can be in this, but we want as many patients. We want them fast. And in fact, one of the nuances of any clinical trial is you get an idea about running an experiment, and then you get bogged down because you don't get as many patients, or another company comes in and wants to enroll patients. And the graph of what you hope of enrollment drops off, and you don't get to your goal. Our goal is 1,000 patients. This program is now, in a little over 1.5 years, has enrolled about 580 patients. I don't know the exact number today. Someone will correct me. And that's 10 times faster than any other ALS trial ever. Now, there are other kinds of diseases that might enroll faster, but this is a select population, really distributed around the country. So it's meeting its goal of bringing patients in. Now, not only, of course, do we collect blood and convert it to those stem cells, we're collecting blood, and we're doing spinal taps. We're having a complete biological signature, and we're storing that. In fact, we already have 27,000 specimens that are available to be done, that are available for research. And something I'll repeat in a few minutes is that this program is not meant to be some academic closed program. And by the way, 5 years from now, we publish the results and the rest of the world gets to see it. It's very open source. These samples are already publicly available to any company or academic that wants it, as well as the clinical data. Of course, it's HIPAA compliant, that is, patients are anonymized. But the point is to make sure, as we evolve, so that others have access to this kind of program. That's the way we believe science should be done. And so the first step in this program is actually to do what's called whole genome sequencing, the actual sequences of the entire genome of our patients, which is done with the collaboration at the New York Genome Center in New York. And again, all of those whole genomes have been done on about 300 patients. They're all being analyzed now to eventually be integrated with the rest of the program. One of the newest things that we face in personalized medicine, now this may be into the weeds for some of you, but it's an important issue. As we're able to understand the entire genome of the patient, some patients are going to sort of say, by the way, do you actually have any other ALS genes? So we probably think we should, since we're going to know that data, we should be giving it back to them. But actually the geneticists in the country believe more. Of all the genes that we can study, there's about 57, 59 genes that if we knew you had that gene, maybe that heart attack risk gene, or that breast cancer risk gene, or some other defined gene they've listed, a patient should know about it. Whether they have ALS or not, you should know you carry this high-risk disease gene. How do you give that data back to patients? It's a research study. How do you make sure it's not me telling someone about a heart attack gene? I'm a neurologist, not a cardiologist. How do you bring in other experts to make sure the right information is conveyed to the patient and or their family? And these are not easy issues to sort out. At minimum, at an organizational issue, that costs extra money. We didn't budget for that. Who pays for that? Does the patient pay for that? It was a research study. Now they got to pay for this other information. Or do you tell them, by the way, we have some other information about your genes, but you have to see someone else. We can't tell you yet because that's the way things are. So that's actually being discussed, and it's not being discussed just in this program, but other programs as well. One of the novel parts of the program is how do we collect data in a novel way at home, not through just Skype, through a call. And so there's a lab at Johns Hopkins, the Cotta lab, John Krakow runs this. There's a motor performance lab, and they're actually ex-Microsoft programmers and game programmers there. And so they've devised this really novel, simple app, not heavy text instructions, where each day a patient tests either a motor function, tracing an object, tapping. In fact, the first one is speech, just collecting speech, because ALS affects speech. You get slurred speech. Can we quantify that? Is it a domain that we certainly know, but we never really quantify? A very important domain is we now know that ALS patients actually develop some amount of dementia. We didn't used to think so, but we now know some ALS patients, not dramatic, but mild. Well, that's a phenotype we don't really capture. We used to think it was just depression. It's not. How do we capture that reliably? Dementia testing, cognitive testing, which is how we detect dementia, it can take hours just for one time. But with the help of IBM Watson, sorry, Microsoft, we're showing them cartoons. And with IBM Watson's linguistics module, when the patient describes those cartoons, the words they use can be used by Watson to determine if they have a cognitive decline. A really novel clinical parameter that we think could be useful. This just points out we generate iPS cells. Those are the cells we start from blood. We convert them actually into the cells, the brain cells, the motor neurons of patients. We've now done this on hundreds of patients. And in fact, 30 of those lines are already available for anyone in the world to take from us and to use on their own. In fact, as of today, we've now, as I said, 580, 560, this is actually about 2 weeks old, the slide. iPS lines are available, 27,000 samples, really available worldwide. Well, the heart of this is once you get all this data, how do you integrate it? Ernest will tell you more about that, but through all of these domains, what's called transcriptome, RNA, DNA, protein, epigenome, in fact, Ernest's specialty, how does the environment change our genomics? You can come up, when you integrate that data with different pathways. Here's an example of actually such raw data. I'm not here to give you a lot of science, but I think most of you can say when I look green, these are actually people who don't have ALS, who have ALS. You can see there's something different here. That's environmental changes in your DNA, known as epigenome. Here's just your RNA, and here is proteins in the cells. You can see there's very big differences. So just integrating all those three parameters, Ernest has already told us that we can define subgroups of patients in this very early study. And these subgroups give us targets for therapy as well. Now, this is not all sporadic ALS. That's our initial attempt. These are examples. This is actually what we call positive control. This is done on a disease where we know the mutation, and it turns out the results, I'm not going to go into the science, but the results of Ernest's integration data matched existing literature done by other researchers around the world. So it taught us that this approach of integrating data can give us the kind of answers we want, at least from familial, but the real challenge is patients who don't have a genetic cause, sporadic patients with disease, happens out of the blue. They didn't inherit it from mom and dad or grandparents. How do we sort that out? So we did a pilot, we just did this. We took almost 30 sporadic patients, again, no obvious cause. And we did some of that very early analysis, not as complete as I just showed you. And lo and behold, we looked at that data, and if you look here, this is kind of red, green, different, these are all ALS patients. There's no pattern there at all. In fact, we only found 2 genes out of the many thousands we studied that were different. kind of disappointing to us. We're kind of hoping to see a signal. However, one of the parameters when we chose those 30 patients is some patients, this is a measure of how people are doing over time. It's a score of functionally how they're doing. This patient isn't changing over about a year and a half. This patient's dropping. This patient's dropping a lot. Here you can see green, very stable. These patients are dropping. So those are what are known as fast progressors versus slow progressors. And how fast the disease is overcoming someone before they pass away. So in that 30 patients, we had fast and slow. When we added that clinical data, not biological data, but we added that clinical data point of the slope, how much they progress, to that data set of all the gene changes. We went from only 2 genes to almost 200 genes that were different. What that teaches us is we don't know the pathways yet. It teaches us that, in fact, clinical data will be a great discriminator of how we interpret our data. It's not just going to be the biologicals. We're going to have that clinical data. And quite frankly, in a program like this, in all programs that we do in clinical and basic science, so it's great to get these ideas. How do you make sure you're right? You have to go back to patients. You have to go back to autopsies and say, yeah, this pathway, it looks correct. I've got to look at real brain patients, other patients, to see, has it really changed? You can actually change things in a fly. You can use fly models to test. So they're known as post-analysis, to be sure, before we think that this is the right drug pathway we have to go over, we got to make sure we're correct. That's a lot of follow-up research, but critical research when we do it right, and we eventually bring the right drug to patients. Here's examples where we're testing things in flies. Flies get corroded eyes when they get an ALS-like gene, and we find genes that fix them based on the results of that kind of an analysis. I'll skip this for the sake of time. The big deal is, how do we actually integrate all of this data? So we have a lot of biological data. We've got clinical data that's coming from the clinical sites, biological sites, and we have to store it. This is where actually Microsoft Azure, Microsoft has kindly actually given us years of, and we hope this goes on for many years, free storage and compute time. I can use the podium to push, get more and more from you. And actually, there's a lot of this program that comes from industry that's, if you will, a pro bono, to use that term, law. And so we have to integrate the data. Ernest is going to tell you a lot more about how you integrate the data. And eventually, not just integrating, coming up with stuff that we in the lab use, how does the rest of the world get to that data? How do they get to see it and query it and get the raw data? And to that, you have to have some kind of good portal, some nice web page where you can say, I want to see the genes that are abnormal in a 50 to 60-year-old person who's male, with ALS, who lived in New Jersey. And so we need that. And in fact, those are pretty complicated portals. And Accenture has offered some of their services to help in integration, in fact, with Microsoft as well. And we have other analytics. And again, Ernest will tell you a little bit more. So in a relatively short period, I know I'm already over, I've tried to give you an example of how we use the general personalized or precision medicine approach that Anthony mentioned to you, Dr. Rosen mentioned to you, and how we apply it in a quite comprehensive way to ALS. We think this is one of the largest, not in terms of number of patients. There are NIH efforts underway to look at 1 million patients, but just for whole genome.